Hello.
Hello.
Yes.
Sir, this is Navin from NPTEL, sir.
Yes, tell me.
Yes, sir. So, who are the TAs, sir, so that I can make them as a co-host?
Should have joined. Is it essential for them to be here?
No, sir.
Is it essential for the TA to be here?
No, sir. No, sir. If someone is there, I can make them as a co-host. That's it.
Right now, I'm not seeing anybody, but that's fine. I'm here. So.
No, you already informed us, sir, them to join.
No. There was only one who was supposed to join. I'm not sure where he is, but we can continue without him for now.
Okay, fine, sir.
So let me know when to start.
5 more minutes is there, sir.
Okay, just let me know when you're ready. I'll check in the meantime if the TA is coming.
Yes, sir. Mm. So, ready, sir?
Yes, I'm.
Yes, sir. We can start, sir.
Okay, so all I need to do is just take any questions from the chat box and on the Excel sheet, right?
Yes, sir.
Okay. Okay. Welcome everyone. This is the first live session of this course for the drug delivery. So if you have any questions, please do type on the chat box. I also have a list of questions that some of you have sent through that Google form. So I'll start from that. By the way, Rajesh is here. If you want to make him a co-host, you can. His name is Rajesh TA. So, anyways, I'll start taking questions from this.
Sorry to interrupt, sir. Rajesh TA, right?
Correct.
Rajesh is your TA, right?
Yes.
Okay, sir.
Okay, so let me take the questions from the Google Excel. So there was a couple of questions from Rosna. And I think Rosna is here also. Anyways, Rosna wanted to know whether we can share the assignment solutions. So Rosna, we are in the process. Actually, we did try to share it, but there's some technical fault. But I think within 1 or 2 days, all the assignment solutions will be shared on the discussion forum. So hopefully that will answer all your questions there. There was another question from Rituja, where Rituja is saying that she is interested in vaccine designing and want to go further in it. And she thinks that this course has helped her, so that is great. She's asking what to do in future if she wants to use this as her career, I guess. So there are few options, of course, depending on what field you are in currently. If you're already familiar and have a degree related to that area, you can always look to either go in industry, or you can also look to further studies such as master's or PhD in that project. Then from there on, you could become an independent researcher, or again look to go into industry at a more senior position. Those would be the two options in vaccine design that I'll suggest. There are also opportunities to link to some funding agencies that fund vaccine research, if that is something of interest to you. So there are a few options. It just depends on what your current academic status is and what is your interest. There's another question from Shreya Tiwari. So Shreya wants to know if I can explain how the adsorption of protein depends on the surface hydrophobicity, and what is the natural state of any protein in the body. So let me take the second question first. So natural state, Shreya, is basically dependent on the amino acid sequence. Are interacting with the serum, or essentially the fluid water aqueous phase. So what generally happens is the hydrophobic moieties will tend to hide from the water phase and will tend to be buried in the protein structure, whereas all the hydrophilic moieties will tend to be on the outer edge, trying to interact with the water molecules. So each protein will have different conformation, again depending on what the sequence is. So that's the natural state of that protein. Some proteins are obviously membrane bound, so in those cases the structure is different because the membrane. So the proteins can expose their hydrophobic regions in the intramembrane domain, and that can then be used to define the protein structure. In terms of your first question, which is to please explain how the adsorption of protein depends on surface hydrophobicity. So basically, since if a surface is hydrophobic, and if let's say a protein is trying to come in contact with that, then at that stage there are chances for that protein to be able to expose its hydrophobic domains, and then those hydrophobic domains will have much more favorable interaction with the hydrophobic surface. So the more the hydrophobic surface is, the more denaturation you could expect in the protein, because all the hydrophobic moieties will tend to then get exposed, and the hydrophilic moieties will then go on the interface with the water. So essentially, you have a surface out of which a lot of hydrophobic interactions are happening, and on top of that, since there is water, there will be mostly hydrophilic domains. So it will essentially bifurcate into hydrophobic and hydrophilic regions. Hopefully that answers your question. I believe is there still a facility for them to unmute themselves also, or this is mostly through the chat or through the Excel? I think, can you guys unmute? Can somebody try?
I think there is an option. Others, I can see that they are unmuting and they can ask questions.
I see. Okay. Okay, then let me just take all these questions that are written here first, and then we can see if anybody has any questions, and we can unmute. Okay. There's another question from Shreya. And again, she wants to know the pattern of the final exam and type of questions that will be asked. So the pattern will be quite similar to what you've seen in assignments, and the type of questions will again also be quite similar. So we will have about 35 MCQ questions and about 15 fill in the blank type of question where you have to put in a range. So let's say the right answer for a question, let's say, is 5. So a range may be defined as 4 to 6 or 5.5 to 6.5, depending on how accurate the answers are in the question. And so yeah, 35 of MCQs and 15 of those will be present in the final exams. And again, the level of difficulty will be similar to what you're seeing in the assignments. Okay. There's another question. Rajesh, you have access to this Excel sheet?
Yes.
Okay, you want to take up the questions? I think there's only 1 or 2 remaining now. So maybe you finish up the questions on the Excel sheet, and then you can take the questions in the chat.
Yeah, I think you were with Shreya's question last, if I'm not wrong.
Yes.
I think the next question is from Sai Prashant KRS. So throughout the course, we talked about the hydrophobic interaction of proteins to the implants. My doubt is that proteins have hydrophilic moiety too. So I could not read after that. So they do need to interact. Sorry. The question is, can you please read the question, Rohit?
Okay. Okay. I'll go ahead and read it.
Yeah.
So the question
I can. So they also tend to interact with implants if the implant is hydrophilic. So then, is there any particular reaction like we studied, like MAC receptor and all? Is the question. So basically, he's trying to talk about the interactions which we have it on an implant. So where if the implant is hydrophobic, I mean proteins have both hydrophobic and hydrophilic residues. And if the implant is sometimes if it is hydrophilic, what will be the interaction? And what kind of receptors basically they will try to interact with is what he is trying to ask a question.
Sure.
Should I answer or?
No, I can answer it. So Sai, generally what happens is you're right. If there is a hydrophobic surface, and I just answered that in the other question, they tend to get denatured and expose a lot more to beams. Having said that, it's not that on a hydrophilic surface there will not be any denaturation. Obviously, every surface is different. So when we say hydrophilic, there is gradation of how hydrophilic things could be. Some could be extremely hydrophilic, some could be moderately hydrophilic, some could have low hydrophilicity. So all of this will essentially result in some denaturation. And again, depending on the protein and the type of amino acid sequence, some of these cryptic receptors sequences can get exposed, as we saw in that MAC receptor example that you're giving. So it is possible that some of that does get exposed even on hydrophilic surface, but the general trend is still the same as we discussed. The denaturation will be more on hydrophobic surface, and hence more chances of cryptic sites being exposed or toxicity with it becoming high. But yeah, overall, any surface will cause some denaturation because it is different from water and whatever the protein has seen before. Okay, I think these are all questions from the Excel sheet. I've already answered Rosna's question. The assignment solutions will be uploaded very soon, so please watch out for that. Rajesh, you want to take questions from the chat before we then ask people to ask their questions directly?
Yeah, I don't see any questions as of now.
There are some questions to me as a direct message, so I'll just directly read them. So there's a question from Sana. That's only the name I see. Rest of it is not fully shown. But Sana wants to know, sir, I have about 10 years of gap of study. Now I want to continue my study and want to apply for PhD through some scholarships. As I've done MSc and have good academic record, I have also written proposal. Is it possible? Yes, of course, Sana, it is possible. You just have to explore various labs that may be interested in a profile like yours, and there are several biology labs. So one thing is to directly approach them. Other thing is, if you think that the fellowship or scholarship that you are eligible for, if you procure that first, then labs are even more open because you come with your own funding. So yeah, do explore, check out which lab has any vacancy, and you can then use that to go. Of course, also depends on your interest if that particular lab works in the area that you're interested in. In this case, I think you mentioned some drug delivery based polymer systems. So yeah, do explore those things. There are several labs. ISC has 3 or 4 that I know of, and obviously every other institute will also have some labs. Okay, there's a question from Sam, which says, sir, can you please explain how did you derive the intoxication level for ethanol consumption of the zero elimination example you gave in the second or third lecture of the course? So these intoxication levels are determined experimentally. So there we had just said a certain concentration, but that concentration people have measured. So if there are cases of alcohol toxicity and they go to hospitals, there the hospital doctors would then measure how much alcohol is present in their serum or the body water, and from that the conclusion has been drawn that okay, above this level some cytotoxicity start to observe. Some of this could also be directly trials, so maybe they might have even recruited patients and gave them some alcohol and seen at what level they started to not walk straight or things like that. So yeah, these are some things that are done experimentally, and those values are then reported, which then others can use for whatever application that they're looking at. Okay, there's a question from Bipin. So Bipin is asking whether this course helps in BE biotechnology. So Bipin, yeah, of course, depending on what your interest and what your future goals are, if your future goal is to do some research in these drug delivery systems, then obviously this will help. If you're not really interested in this, then it just becomes informative. This may not be the right course for you. So yeah, it just depends on your interest. But yeah, if you're definitely interested in any kind of drug delivery systems, then this course will give you a very, very comprehensive overall background on what different systems are and how they're used in clinics and in research. Okay, there is a question from Shreya. She wants to know that is it possible for them to join my lab? This just depends on several factors, Shreya. From time to time, there are vacancies, and this will be posted on the website. You can explore that, but yeah, let's keep that platform for general questions and see if others have anything. But yeah, we do take people from various different backgrounds, such as chemical, like you mentioned, along with everything else. Okay, I think that's all the questions I have. Rajesh, is there any question that was sent to you directly?
No, Rachit, I don't have any questions.
Okay. In that case, if people have questions they want to ask, they can maybe raise their hand or somehow indicate that they have a question. I think if you go to reactions, there's an option of raising hand. So do go ahead and raise your hand if you have any question, and we can answer that. Okay, I'm not seeing anything. If people want, they can directly unmute themselves and ask if they're unable to find this hand function. Okay, we have maybe we'll wait 2-3 minutes and see if somebody wants to type or ask a question. Otherwise, we can finish with this live session. So yeah, if you have something, please either type or speak up, and we can take that over the next couple of minutes.
Rachit, there is a question from Priya.
Okay.
Which synthesis process are good for increasing the porosity of nanoparticles, specifically metal nanoparticles like copper or basically copper sulfide?
Okay, so I'm actually not very familiar with the copper sulfide particles. Generally speaking, there are some sort of etching that is used to make things porous while things are being formed. So this could be acid-based etching, or this could be some sort of seeded growth in which things are encapsulated and somehow leached out. But with metals, I'm not very familiar with this. If you send me an email, I can try to get back to you after reading a little bit on this. By the way, Rosna, we have a quick update. We have figured out what was the problem in terms of uploading the assignment solutions. So it should be done by the end of the day today.
I think the solutions are for the last 5 weeks. It has been uploaded.
Yeah, but they were somehow not live. Kamini just
Okay.
told me that there was something
Sure.
she needs to do. So
Yeah, we'll do it today.
There's another question to me, which is what is the procedure to join the Mtech program at ISC? So there are several Mtech programs, especially in engineering departments. So the procedure is to directly apply when the applications are open. So ISC's application opens twice a year, once during the during November interviews. So I think the applications open sometime early October. And then once they are open in March, April for interviews in May. So you can apply to those Mtech programs if you're interested. Priya, send me her email ID. I'll note it down. I'll see if I can get your answers to the porous metal particles.
Yeah, Rachit, there's a question by Rosna.
Yeah, go ahead.
I have hands-on experience on using gold nanoparticle synthesis. Can I use it for drug delivery?
Absolutely, absolutely. Gold nanoparticles are used quite a lot, both as contrast agents for imaging as well as for photothermal therapy. So they can themselves act as drug. They heat up under light or under some ultrasound. So wherever they are accumulating locally, they can cause tissue damage. So if they accumulate in tumors, you can use that to sort of hurt tumor cells. Sometimes the drugs are coated, absorbed on top of nanoparticles, and then they are used for delivery as well. So there are various applications of them. There are actually very nice reviews out in the field that you can refer to for basically comprehensive idea about how some of this is used. There's a direct message to me from Sana here, who's saying that she has enrolled for the course of NPTEL but unable to give exam because she's in Qatar. Yeah, I mean, you can definitely. So she's asking, she cannot give exams this session, but she may be able to give it in future sessions. So can she enroll again? Yes, Sana, you can definitely enroll again for a course if you would like to. question by Shreya to me, which is what is meant by high polydispersity, which is a disadvantage of emulsion-based methods. So essentially what it basically means, so let's say I want to make 100 nanometer particles, but I use an emulsion-based method. I know a speed in which predominantly most particles are going to be 100 nanometer. But what happens is, let's say here's a propeller that is spinning and giving the energy to the solution. So whatever particles that are forming and closer to the propeller, they get a lot more energy. Whereas whatever particles that are away from the energy source, let's say towards the edge of the beaker, get lesser amount of energy. And as a result, you get a particle distribution. So smallest is right next to the propellers and largest particles are further away from the propeller. And that's why emulsion-based methods tend to be quite polydispersed. And that's basically what is polydispersity. So distribution, how dispersed the distribution is, how maybe what is the standard deviation, how high that is. That's the measure of polydispersity there. So that's why it's a disadvantage, because obviously if I'm trying to make a certain size, I want 1 micron or 100 nanometer, I would like all my particles to be there, because it may be possible that if I want 100 nanometer and if I get a 500 nanometer particle, that will not perform the function as optimally for that particular application. Rajesh, you want to take that Ritam's question to everyone?
Yeah, is there any provision for 6 months internship program from IAC or MSc dissertation? If so, when and how to apply?
There is, Ritam, you can directly approach the labs that you are interested in. If they have vacancies, they will reply to you. Yeah, but generally there's no central procedure. You just have to directly approach individual labs.
Hello, sir. Am I audible?
Yes, Ritam.
Yes, sir. Should I be mailing like individually, like after researching like which faculty to approach? Should I be mailing the faculties personally or the HR or something?
No, mailing the faculty is what I would recommend.
Okay, sir.
But again, it depends on whether they have vacancy or not.
Sure, sir. Thank you, sir.
Okay.
There is one more question from Sai Prasad. He says he's from a zoology background, and the kinetics questions, a part of the kinetics was a little bit tough for him. So for the final examination, do we need to study the equations and all in deep? And I think the same question goes to Priya also. Priya is like the kinetics part was a little bit tough for her.
Yeah, so the maths in this course is actually quite minimal deliberately, but I do understand, depending on the background, some of you may find it tough. So in terms of the equations, I will give you the equations in the exam. So I will not ask you to remember the equations. The equations will be given to you. This will be a sheet of paper or a separate attachment on your online exam. But you will still have to know how to apply the equation and which equation belongs to which sort of drug release. So those are some things that you would have to at least understand that part. And I'm sure if you understand the course very well, and if you have questions, ask TAs and do the assignments, some of these will be quite not very difficult for you to do. Okay. I think that's all the questions there are at
Sir?
this point. Yes, who's this? Could you please identify yourself and then ask your question?
Sir, Rosna here.
Yes, Rosna.
Sir, you mentioned during the exams, when we write the answers, we need to mention the specific range of if you get 0.5 is your answer. If you mention like 0.221, so will it be considered as a correct answer?
No. So you have to give a single answer. You don't have to mention the range. Range is what the software will check it against. So what I'm saying is you don't have to be absolutely exact. So for example, if the answer to a solution that you find is 5.02 and so there'll be a range which will be given in the software. So if anybody answers 5.03 or 5.01, all of those will be correct. So that's what I meant there. But for you, you will only have to answer a single number.
Sir, one more thing. During the equation solving, so do we need to solve it like stepwise, or we end up just mention the answer itself?
Just mention the answer. Where you of
don't
course
have
to get to the answer. Yeah, to get to the answer, you'll have to
yeah, of course.
basically do the steps. But in the software, you can only enter just the final answer. You don't have to enter any of the steps.
Okay. Okay. Thank you, sir.
Okay. Another question from Shreya, which is, where can we find the solution to the assignments? This will be posted very soon. It's not posted there yet. It will be posted by the end of the day. So TL will make an announcement. Rajesh, please, once the things are live, you just please make an announcement.
Sure.
Excuse me, sir.
Yes, Bipin.
Final exams are pen or paper mode or online mode, sir?
They're all online mode.
Okay, sir. Thank you.
Okay. We'll wait another couple of minutes to see if there's any other question. So if you do, please either post it in the chat or raise your hand or directly unmute yourself and ask.
Hello, sir.
Yes, Preeti.
Yes, sir. So good evening. Myself, Preeti. Sir, I'm working on drug delivery. Actually, I'm a PhD student and in third year. So I have one question that when we do the drug release study in vitro, like drug release, so in most of the paper, they have mentioned that we withdraw some amount of the stock volume for to study the release, and then put the same volume to restore the whole volume. So storing the same fresh buffer in the batch, it doesn't really change the concentration of drug. So why we put back the same withdrawal amount with a fresh buffer? Rather than calculate or do some calculation.
Yeah. So this is interesting, Preeti. So there are lots of different ways people do it. One way, of course, like you mentioned. So what they will do, so just for everybody's perspective. So let's say they make a scaffold and they want to see how much drug is coming out. So what they'll do is they'll dip that scaffold in, let's say, 10 ml of a solution. And every time point, maybe they have 10 time points that they want to study it at. So let's say every hour. So then every hour they will take out some 100 microliter or whatever may be required for their assay. And then what they generally do is they'll put 100 microliter buffer back. So that's one way to do it. Of course, it depends on how they are measuring the drug. Sometimes the technique that they're using, that 100 microliter that they've withdrawn would be basically gone in the technique. So if that's the case, then that's when most people then prefer to then make the volume up. And the reason they want to do that is so that at least there is equivalent amount of water throughout the study. Sometimes people don't do it. Sometimes people say that, okay, 100 microliter is almost negligible in front of 10 ml. And so then they'll just take it at 10 different time points. And finally, the volume will reduce from 10 ml to 9 ml. And they can adjust the calculations accordingly. It becomes difficult to do that. But so all of those methods are well acceptable, Preeti, as long as the volume that you're drawing is much smaller compared to the total volume.
Is one doubt is that this, like if you have a large volume like 500 ml or 1 liter, then it is kind of negligible the amount we withdrawn and restored. But if you do the experiment in a small volume like 10 ml, then it is a considerable difference it will create.
Correct.
Then
Correct.
I have posted a question several times in the ResearchGate on the ResearchGate. They have said that do the dilution factor, add the dilution factor. Then if we do for 1 day, like, and withdrawal several times in half hour step. So there is a lot steps. There are many steps like 20 or 30 times, then it will create a huge difference in the volume itself. Then for a small volume, how can we tackle out these things?
Yeah, no, it's challenging, Preeti. So one way to do it, if the sample is not extremely expensive, then you can even think about having 5 of that setup, and you use the first one for the first 2-3 time points, the next one for the next 2-3 time points. So you can go sequentially like that. Otherwise, you'll have to somehow calculate the dilution that you're doing when you add fresh buffer instead of the original sample.
Okay.
So these are experimental limitations that one has to face.
Okay. Thank you so much, sir.
No problem. Looks like there are a few questions now in the chat box again. Rajesh, you want to start taking them?
So the first question is from Priya. What is the ideal size of nanoparticle? Like below 6 nanometer is good for clearance, but coating and functionalization leads to increase of size of nanoparticle.
Yeah, so interesting question. It again depends on the application, I would say. So like Priya mentioned, if the particles are below 6 nanometer, generally kidney is quite good in clearing them. So, I mean, if your application requires the particles to be cleared rapidly, then you would want to be below that range. But most applications of drug delivery you would want sustained release and much longer residence in the body. So in those cases, your particle size range should definitely be above 6 to 10 nanometer. There's obviously an upper limit to it. If you're going, let's say, IV injection, intravenous injection, then you can't be beyond, let's say, about 5 micron, because your blood vessels, the smallest capillaries are about 5 microns. So then that sort of limits the size range. Then your immune system is also an important player here. They take up particles quite efficiently in various size ranges. So you have to be careful on how much size is there. For example, liver and spleen will clear things away very quickly. Things that are above 300, 400 nanometer. So generally speaking, if you're doing IV injection, you would want to be between 10 nanometer to about 200 nanometer for longer circulation. But again, it depends on application. You may be directly going intramuscular or subcutaneous, and then these ranges will change. Okay, before we take chat question, I think there's a hand raised. So Asad, you want to unmute?
Yeah, hi, sir. Sir, I just have a question. Is there any physical property of a cancer cell that we can use for targeting our drug vehicle? Like physical property in a sense, any change in that cell that can interact with a polymer-based vehicle. So is there any kind of physical property? Okay.
Yeah. So there are a few, Asad. I think actually towards the end of this course, we will cover that topic. There's this thing called Epr Effect, which causes the blood vessels to be much more leaky around the cancer cells, because these vessels are quite immature, and so if you have, so there are larger gaps between the cells, and so let's say a 50 nanometer particle which may not come out from a healthy vessel will come out in regions where tumor is located because there are gaps. There are other receptors, not ideally physical, but there are chemical things that you can do to target tumors also.
Like we are using a polymer-based vehicle, so is there something, any kind of property that can attach a polymer-based vehicle?
Yeah, so you can attach ligands to your polymer. You can make the polymer of a size range which will use this Epr effect that I just talked about, which will be covered in a lot more detail in the course. Those are the two most widely used sort of strategies. There could be others that will then depend on the specific tumor and specific sort of condition that one is looking at.
Okay, sir. Thank you.
Okay. Going back
by Ritu. So, sir, I have missed the last date for exam registration and I want to register now. Is there any way? Please suggest it.
Okay, so this would have to be directly emailed to the NPTEL folks. So if you email them, they may tell you whether it's possible to register this time or would you have to just take it in the next session. So just directly email the NPTEL contact who are handling the administrative part of this course. Okay, there's a question directly to me, which is, can I share my email ID for future reference? Yeah, sure, please. It is publicly available on my website also. This is Rachit, R-A-C-H-I-T. I'll just type it here actually. Again, I don't see anything on the chat right now. Okay, Asad has another question. Go ahead, Asad.
Yes, sir. So there are a number of possible drug delivery systems, but can you just comment on the feasibility of a system for commercial application? Because so far is there from all types of commercial vehicles applicable. So can you just share something about feasibility? How what can be done in future?
Feasibility of nanoparticles, is that what you're asking?
Feasibility of these drug delivery systems. How much significant they are?
Yeah, so again, this is something that we will cover in the course. We have covered some already and we'll continue to cover some more as we go. So the feasibility is quite feasible, I will say. There are at least, to my knowledge, there are at least 30, 40 commercial products that are currently being used in clinics and patients, which use a lot of these concepts that we are learning in the class. You mentioned about tumor targeting. There are things like Doxil, which is a liposomal doxorubicin. Again, something we'll cover. There's Abraxane, which uses paclitaxel as anti-cancer drug with albumin. So all of this we will cover in the course. There are other depot sort of system for contraceptives that are also used. There are several, and actually more and more research is going on. There are a lot of clinical trials that are going on these aspects. So in that sense, I would say that this is quite feasible.
So I'm just working on one of the systems. Means I'm trying to make something out of that. So I would like to discuss you in the future. So I will share some of the document on your make.
Sure.
That would be great if you can just. Thank you, sir.
Sure, absolutely.
Thank you.
Okay. Again, we'll give a couple of minutes to see if there's any other question before. If not, then we'll end this. So if you have anything, please feel free to ask.
Sir.
Yes. Yes, Luzna.
Rosna here.
Yeah.
Sir, what is the like the best advantage we can say about when we talk about long-term medication? Generally, doctors say it can harm your kidney and liver. So this has any advantage? Because compared to intravenous injections and oral tablets, this targeted injections like all these nanoparticles or hydrogels, whatever, does it protect your kidney in some way compared to a drug oral drug and injection?
Yeah. So it does, Rosna. It does a little bit. Now, the extent of it depends on how much. So there are two things, right? If you remember that curve that we had shown about multiple administration and sustained release, where one curve was going up and down like this, and another was basically that. So if you look at that, basically what we're doing is to maximize the time of drug in therapeutic range. We are giving very high doses, even sometimes breaching the toxicity level. And then it obviously drops down as it's cleared, either metabolized or excreted. So those things are not good, especially for toxicity things, because if they are having some effect on kidney or liver or any other organ, then those things will get amplified with the dose. So having a more sustained profile will help. Having said that, it may still have toxicity. So it helps in reduction of the toxicity. It also helps in sort of patient compliance, because let's say if I take something and I feel like vomiting, next time I have to take the tablet, I'll think twice before taking it. But if it's already sustained release, I've already taken it once, then at least the patients, even though they may still feel vomited, at least the drug is still doing its function. And so the patient compliance goes up quite a bit. Toxicity does reduce, but it depends on drug to drug and what sort of toxicity levels are being reached when you give IV or oral dose versus the sustained release formulations.
Okay, thank you, sir. Sir, especially this comorbidities, those who are on continuous medication for cardiological problems and diabetic and all. So if this could reduce for 5 to 10%, of course those things, definitely when they are on more than 10-year to 15-year medication, definitely they'll end up with a kidney issue or a liver issue. So that's what I was.
No, very, very relevant, and in some cases it does help, depending on application to application. Yes.
Okay, sir. Thank you.
All right. We'll wait just one more minute to see if there's any other question. Okay. In that case, thanks a lot for everyone in participating in this. It's always a pleasure to directly interact with the people who are taking this course, and please continue to have discussion in the discussion forum. The TAs are there to help you. If you find there's some things that are not answered, please do directly email me, and I'll make sure you get your answers. Thanks a lot, and I think we can probably end this at this stage.
Okay, sir. Shall we wind up?
Yes, please.
Okay.
Thank you, sir.
Thank you, everyone.